Welcome to the North American Fruit Explorers 2024 virtual conference. My name is Sean Charles Williams. I'm a director on the board of NAFEX, as we call it, the organization for short. And I'll be serving as a facilitator for this session. This session is Vitis Vinifera and Muscadines, grape breeders seeking the best of both grapes. And first, this is in a webinar format versus what you might be used to, which is a meeting format in Zoom. So you won't see any picture boxes except for just the speakers and myself and Chris O'Mannix, who's also a co-host. And secondly, you'll notice that we're using the Q&A section for questions versus a chat. So if you have a question for our speakers during their talk, please put it in the Q&A section so we can address or feed those to the speakers as they're finishing up with their presentation or they conclude their presentation. Second, we encourage you to ask questions, like I said, via the chat, but also, finally, this webinar is being recorded and made available to our members and conference participants. After the conference at nafex.org, you'll find this content along with bonus resources like printable versions of our speakers' presentations. And just a few words for NAFEX, for our new members, this might be the first time that you're joining us. NAFEX was founded in 1967. The North American Fruit Explorers is a network of individuals throughout the United States and Canada devoted to the delivery, cultivation, and appreciation of superior varieties of fruits and nuts. NAFEX members share ideas, information, experiences, and fruit propagation material via our website, social media channels, fruit-specific interest groups, and annual conferences like this one, whether in person, online, or a combination of both. As a paid NAFEX member, you get 4 editions of the Pomona Journal each year, as well as the ability to access past Pomonas for up to 50 years, which have been digitized and are in our digital library at nafex.org. So there's a wealth of fruit growing information in there, as well as information if you're a fruit explorer or just trying to figure out what has been done in the past and what you might want to do in your own efforts. If you haven't already looked at the website and all these resource links, we highly encourage you to do so. So with that, it's my great pleasure to introduce 2 of our speakers today. They are, let me get out of the screen view here. They are Renee Threefall and Margaret Worthington. Renee is a research scientist with the University of Arkansas Food Science Department. She's a leading authority on U.S. fresh market juice and wine grapes with over 25 years experience working on quality, sensory, and post-harvest evaluations. She has published 58 journal publications and 7 extension fact sheets. She was the recipient of the Julian Miller Senior Distinguished Researcher Award for the South Region American Society of Horticultural Science and is the administrator for the American Society of Enology and Viticulture and Enology. Oh, we did that twice. For the Eastern section, she has extensive experience in leading and co-leading grant projects. Our second speaker is Margaret Worthington. She's an associate professor of fruit breeding and genetics at the University of Arkansas, leading applied blackberry, grape, and muscadine. It's not muscardine, it's muscadine breeding programs. She worked with private industry in California to develop uniquely flavored grape and muscadine hybrids. She has published 50 publications in the past 10 years, including the first dense genetic linkage map of muscadine and the I'm not even saying phylogeny of wild and cultivated muscadines and a candidate gene for berry color in muscadines. In 2024, she will publicly release the first 2 University of Arkansas muscadine cultivars. So we might get to hear about those today. So once again, please put your questions in the Q&A section. And with that, I'm going to turn it over to Renee to do the first part of our presentation. And then the second part will be, I'm sorry, Margaret will be the first part of our presenter. And then I'll turn it over to Renee. So Renee, I'll ask you to go ahead and share your screen.
You want me to share my screen, I think.
Margaret, yep.
You see it?
Yep, it looks great.
Okay, great. Well, thanks very much for inviting me and Renee here today. Renee and I are working very closely together in the horticulture and the food science department here at the University of Arkansas Division of Agriculture on our favorite crops, grapes and muscadine grapes. So, what I want to do to start us out today is give kind of an outline. I'm going to talk a bit about what are muscadine grapes first for those of your members who might not be familiar with them. Then I want to give a little bit of background about our grape and muscadine breeding program here, and then a little bit of the history of breeding these wide Vitis by muscadine hybrids. And then I'm going to turn it over to Renee, who's going to talk about our really exciting new specialty crops research initiative project, and the new work that we have coming forward. So, what are muscadine grapes? Well, they are the first cultivated grape in North America. Here you can see a picture of the mother vine of Scuppernong that's still growing. It's the original Scuppernong bronze muscadine. It's in Manteo, North Carolina, and it's this big old ancient grapevine. They're native to the southeastern US. So here in gray is where you see that this region is kind of where muscadines are native to. And here you see this red star, and this is where we do most of our work, kind of near the both the northern and the western extremities of where muscadines grow naturally. So they are not very cold tolerant below 10 degrees Fahrenheit. So it's a great fruit crop to explore for you, those fruit explorers in the southeastern US, but maybe not those in the north. You probably won't find it. Um, let's see. The center of diversity really is here on the East Coast. I had a grad student, I'll talk about this a little bit more, who did kind of a collection of wild muscadines and understanding their genetic diversity. And it's interesting to go look for them in the Eastern North Carolina. They're the dominant understory plant species there. So formal breeding of muscadine grapes began as early as 1907 in North Carolina and Georgia with a fairly narrow set of germplasm, a few wild selections they started this whole breeding effort for. They were breeding for the wine and the fresh market, and a lot of emphasis was placed on muscadine grapes, and then Prohibition hit and the emphasis shifted back a little bit for a few years, and now it's coming back strong. So they are a large fruit. Oh, here we go. Here's where these kind of original founders were found. Most of them in the coastal plains of North Carolina is where they found these original wild plants that they started all these breeding programs. It's a large fruit with an intense fruity aroma and flavor. To me, they are my favorite fruit. I grew up in Eastern North Carolina. I would have them every fall at the farmers market, at roadside stands, and even in the supermarket there. And I think they're the best tasting fruit and grape that there is. I'm obsessed. And so I'm excited and really grateful to have the opportunity to work with them now in my career here. Um, but they do have some things that hold back their approval, like the thick leathery skin and gummy flesh and large bitter seeds. So we're working on improving all of these texture attributes. So, this is not an interactive format, but I have a question. Who here has tried them? So maybe we'll do a survey at the end when we're done with the seminar to kind of talk about other people's impressions of them. So, um, how are they related to grapes? Basically, both muscadines and Vitis vinifera grapes, the kind of grapes that you're used to eating as a table grape, a wine grape, and a juice grape, uh, they diverged about 20 million years ago. They're both in the family Vitaceae. Some people call Vitis and Muscadinea two separate genera, and some people say that they are subgenera within the same genus Vitis. Lumpers and splitters, y'all know who that is. It doesn't really matter. They're fairly far diverged. The main thing that genetically that is different between them, the genomes are super similar, except that there are two chromosomes. There's a difference in chromosome number. So muscadines have a base chromosome number of 20, whereas bunch grapes have a base chromosome number of 19. So there's one chromosome in grapes that is split and basically segregates as two independent chromosomes in muscadine. And It's exactly the same situation as a horse and a donkey or a human and a chimpanzee. So they're pretty similar, but they're diverged long ago. There are some important morphological differences between them. The cluster size is generally much smaller on muscadine grapes, but you can have very big fruit size. Uh, for those of you kind of looking and wondering, is this a wild muscadine or a wild grape out in, as you're doing your explorations, I think the easiest key for me is the tendril architecture. A muscadine has a tendril that doesn't fork, as opposed to a grape that tends to have a forked tendril. So that's a good way to tell them the difference. There's some differences in bark texture as well. Muscadines tend to have a smoother bark texture compared to grapes that have a shaggy bark, and they also have fruit lenticels on them, and that can something that distinguishes them. So they were basically domesticated independently of Vitis grapes. The breeding programs have operated largely separately. So I guess, and here you see, this is kind of a phylogeny. Here you see there are like 50 different grape species or 50 different grape species over here, from the Near East is where Vitis vinifera grapes came from, so like Mesopotamia. Um, but there are wild Vitis grape species in North America, like Vitis labrusca, Vitis estivalis, Vitis arizonica, a lot of grapes that have been used in breeding programs. But rotundifolia grapes, the muscadine grapes, are quite actually distinct and separate phylogenetically. So I'll shift gears now to talking a little bit about the Arkansas fruit breeding program. So our fruit breeding program has existed since 1964. It was established by Jim Moore. He was an Arkansas native, kind of went and did his PhD at Rutgers and was at the USDA in Beltsville for a number of years before he returned. When he came back, he started breeding programs on a lot of different crops. Too many crops. Crops like strawberries, apples, blueberries, peaches, blackberries, and grapes. And we still have active breeding programs in a number of those, particularly blackberries. Those of you who have heard of Arkansas fruit breeding may have heard of us through our blackberry breeding program, and we can talk about that in questions or any other time y'all like later. But today we're going to focus on grapes and on muscadine grapes, which were added in 2007 by John Clark, who was my predecessor in the program. He assumed program leadership in 1997 and retired in 2023. So a little bit about our program. Uh, our research station, virtually all our work is done at the Arkansas Fruit Research Station in Clarksville, Arkansas. We've got about 10 acres in vineyard there. We get about 1,100 millimeters in rain a year, so it's a fairly rainy environment. We can get very, very cold. We got to negative 15 Fahrenheit in 2021, but we can also be very, very, very hot over the summer. We get a lot of chill hours, so we're a high chill environment, at least 1,000 chill hours, although it will never get cold this fall, it seems. We have a host of fungi and insects and challenges working there, so it's a good place to breed for disease resistance, but we don't have any Pierce's disease where we work. It's a little bit too cold for Pierce's disease. So we've had the grape breeding program since the program was established in 1964. It used to be our biggest program years ago, and then blackberries kind of became bigger. We had a number of objectives to bring in these kind of muscat flavors. So muscat flavor you get from Vitis vinifera wine grapes. It's that kind of linalool floral flavor, foxy flavor that comes from methylanthanolite. That comes from Vitis labrusca, those Welch's grape juice Concord grape flavors, and bringing that into a texture that is more palatable, more acceptable to people, a kind of table grape texture with that crisp flesh, thin skin. We've also had a lot of focus on fun sizes and shapes, like those witch's finger type grapes, winter hardiness, disease resistance, and the overall arching theme was to develop an Eastern table grape industry. That was the dream of Jim Moore. So our parental germplasm came from a bunch of French American hybrids, a bunch of selections from the Cornell program. Here are some examples: Alden, Schuler, Delight, Lady Patricia, etc. We had a lot of important founders, and Robert Dunstan selections. For those of you who are familiar with his work, he was a hobbyist breeder in Greensboro, North Carolina. One of the three Bobs, Bob Zender, Bob Farrer, and Bob Dunstan, who are all private breeders, kind of backyard breeders who made really important contributions to grape breeding. We also had material from Meridian, Mississippi, and the Illinois program. And a lot of initial germplasm also came from Fred Huff's program at Rutgers. So we've had 18 public releases, um, a bunch of table grapes that have really great flavor and texture. Also some new wine grapes, and I'm sure Renee could talk more about our wine grapes, although we're focused really on kind of the muscadine and muscadine hybrid side today. Um, but about that Eastern table grape industry, that never has really come to fruition despite our best efforts because it rains here. So it's very hard to compete on a commercial scale with that Bakersfield, Southern Central Valley industry in California where you don't have rain and you don't have these problems with cracking. So that's why John Clark made the decision to kind of shift our focus away from so strongly focusing on table grapes towards muscadine grapes, the initial original Eastern table grape. But we also have had a lot of success in our grape breeding efforts through our collaboration with the private sector. So we had a breeding agreement with IFG that began about 20 years ago. And from that, about 19 products have been developed, including cotton candy. So cotton candy has its flavor from the Arkansas parent, which had a combination of those foxy and muscat flavors that gives it that kind of burnt sugar, interesting flavor. And also gumdrops, drapes, a bunch of the candy series that comes from IFG is derived from our products. And we continue to collaborate with partners in the private sector in California on grape breeding. But our muscadine program has been going since 2007. Since then, we have planted over 20,000 seedlings. I need to update this slide because I meant to do that today, but I'm sure we have more than that. We are up to over 400 selections now, and a group of advanced selections moving along. We were expecting to make our first selection or releases this year, but actually, I think they're delayed until next year due to some cold damage in the initial seedling stage at the nursery. So they'll be out this time next year and available to order. We've had an emphasis on improving consumer quality while improving cold hardiness because we're kind of at the northern extent of the range and also disease resistance. And we have a recent focus on seedlessness and developing wide hybrids. So something that I think is important that we have learned from our experience working on Vitis grapes is that flavor is a strength. So the conventional wisdom for so many years was that people wanted in a table grape something that was sweet and neutral. And I think that our experience working with IFG and developing products like cotton candy has really turned this on its head. Now we know that people are really excited about bold flavors and that these can be wildly profitable and sold at a premium. So Jack Pandal, who is the manager of Grapery, who is the exclusive licensee of IFG cotton candy grapes in the US, had put his email address on the bag of fruit, and he has gotten lots of comments about people's experience. And he said that he's yet to have one comment on berry size or shade of color. People are very focused on flavor and texture. It's all about the eating experience. So what I have taken from this kind of story is that people are excited about bold flavors, but weirded out by new textures. So we really need to get this muscadine flavor that is so awesome. I mean, it makes my car smell like flowers and just wonderful after I carry fruit back from Fruit Research Station to Fayetteville, where we live. Um, but we need to get this into a texture that is more similar to a grape, and we can do that. So currently muscadines are a bit of an outside fruit. So I have here, little rocking chair on the porch and your spittoon here. A lot of people, when they eat muscadine grapes, will just kind of suck in the flesh and not eat the skin and not eat the seeds. And that is not the recipe to build an industry. We want there to be no spitting involved, a sophisticated indoor fruit. So we need the skin to be edible. I've had muscadine skins described as shoe leather before, but that is not always the case. There is a lot of variation within muscadine germplasm in terms of what that skin is like. We have skins now that are much, much thinner than the older ones, that are very adherent to the flesh and much more similar to what people would want from a table grape. I've heard grad students describe the flesh texture of muscadines as oyster, snot, or eyeball-like. So they would just be flying off the store shelves with a texture like that, right? But we also have a lot of improvements in the muscadine flesh texture, and I think it can be a really nice experience now with some of the new cultivars. So we've made a lot of improvements there, but there is one more hurdle, and that's seedlessness. Oh, here you can see one separated with its big old skin and flesh on the inside. So I want to talk more about seedlessness when we talk about breeding wide hybrids, but seedlessness, stenospermic carpic seedlessness in grapes, it's a trait where you actually have to have fertilization occur and the embryo starts to develop and then it aborts during the process. So it never becomes a full hard seed. People can do embryo rescue to breed table grapes from seedless crosses. But it's important compared to parthenocarpy and useful because that seed, the developing embryo actually sends hormones to make the fruit bigger. So stenospermocarpy is a trait that has existed for thousands of years in table grapes that was naturally selected, and it has never been discovered independently in muscadines. So we had to introgress it from muscadines. So we have been breeding with seedless material, and I'll talk a little bit more about how this emerged since 2017. Now we've made, a close to 50 selections over the years, seedless selections, and it's probably 30% of our program right now. I really am excited about the material. I think they're really good. Here's the first seedless selection that we made. It was selected by a bird also who put its nest in our little vine. And we also have molecular markers now that we can use to screen our populations to ensure that we're selecting only perfect flowered seedless material, because we also don't want females that are seedless. That's a dead end for us. We also have a processing breeding program. We work with Renee on things like productivity, uniform ripening, dry scar, cold hardiness, disease resistance, stable color, and we have to make products. So it's a lot more complicated to breed wine grapes than it is to breed table grapes because you actually have to go through and make wine and do consumer sensory evaluations of those products. So Renee can talk more about that process later on when we have our discussion. Another goal for us is understanding cold hardiness. Muscadine grapes are assumed to be cold hardy to about 10 degrees Fahrenheit, although we have seen many selections do better than expected when it gets colder than that. You see things like this like weird orange fungus algae growing on the vines. It's a saprophyte that comes from the kind of sugary juice that's leaking out of the vines after cracking. We see things like aerial roots that come on after cold events, and we end up seeing death of whole canes sometimes. From this vascular damage. So you can see here, there's a lot of variation. This is in 2019. We had a fairly cold winter, and Carlos had no damage at all. Nesbit had a little bit of damage. Supreme and Black Beauty were hit very hard. So, depending on the cultivar of muscadine, there is a lot of variation. We had a very cold event in 2021. It got to negative 15 Fahrenheit in Clarksville, negative 25 in Fayetteville. And this is when all those pipes froze in Texas. Y'all may remember that on the news. And we had about 85% of the vines in Clarksville killed to the ground. So it was a very big selection opportunity for us to find those that did survive, including some of the new selections that we hope to be releasing in a few years. And I will say Carlos has been really a standout for us. That's the number one processing bronze that's grown throughout the country. It does really well in terms of midwinter hardiness for us here. So it's interesting in grapes, the real limitation in terms of cold hardiness tends to be bud hardiness. So you have buds that are killed. We've been working with a former grad student of mine who's now at Cornell, sending him buds and vascular tissue for these cold hardiness trials. So here you can see in blue AM70, one of our new releases, and Carlos, which are kind of considered gold standards for cold hardiness. And then in the red, we have Jupiter and Reliance table grapes, which are both considered to be very cold hardy Vitis vinifera and hybrids. They've got some of the more cold hardy wild Vitis species in there too. So they're basically equivalent in their bud hardiness. The difference is the stem hardiness. So muscadines tend to be a little bit like an irrigation system that doesn't get turned off for the winter. So they tend to have poor vascular hardiness. So the vascular tissue freezes and thaws throughout the winter and causes these cracks that introduce trunk diseases and cause them not to be able to move water when they're trying to produce fruit and transpire in the next season. We also are working on disease resistance. Muscadines are very resistant to most of the major pathogens in grapes, but you see black rot, bitter rot, macrophoma, ripe rot, and even you can sometimes see a little bit of powdery mildew and skin cracking on some of the newer selections. You see powdery mildew more on the actual berries and less on the leaves in muscadines. Other objectives, we're breeding for dry stem scar. That's very important for post-harvest. Interesting shapes, big cluster size, and fun leaves and berry size. Here's some giant ones. You have to get a knife and fork. We also are focused on injecting a little diversity from wild muscadines. As I said, most muscadines were in modern breeding programs came from just a very few selections taken from Eastern North Carolina. So we did this big phylogeny study with a former grad student where we looked at how genetic diversity of muscadines is partitioned across the US. And we really found that most of the genetic diversity, there's bigger differences when you get closer to the East Coast. The material in the West is not as different from each other. Like Arkansas material is pretty genetically similar to Tennessee material and even Texas material, but none of that material has really been employed in breeding programs. So we are making crosses with wild materials from the Ozarks, hoping to bring in some better color stability, some better cold hardiness, so on and so forth. So when are the first releases coming? We hope to make them in fall 2025. We have two that are coming out. The first is AM70. It's a black fruited, perfect flowered selection. Consistent standout for vine health and hardiness for us here. It survived that 2021 Arctic vortex, a Christmas freeze in 2022 with very minor spur damage. And it seems to have a really balanced crop and vigor, so it doesn't overcrop. It's a nice fruit. We have done comparisons of it and Supreme, which is kind of considered a gold standard for quality in a muscadine grape. And it's very comparable to Supreme, a big berry, high bricks, good dry scar, and a full kind of traditional muscadine flavor. Perfect flowered as well, so you don't need a pollinizer vine. AM77 is the other one that we're going to release as a juice and wine grape. Renee has been making wine and juice for a number of years, and we have put it in lots of tests with consumers, and they seem to like it. It has a smaller berry, as is typical, and a texture that is more old school muscadine texture, but people find that the cold hardiness is very well, and it's a nice, vigorous vine that produces well in a lot of environments. So back to wide hybrids. I'll finish this up and turn over to Renee. There's a quote that I love from Harold Olmo, who was the grape breeder at UC Davis, and he said, "Of all the wild grapes, the species rotundifolia possesses the most remarkable combination of desirable characteristics to counterbalance the deficiencies of the vinifera grape." And I think that is so true, that there are so many things about muscadine grapes that would help benefit the Vitis industry and vice versa. There are so many things about Vitis grapes that would really benefit muscadines if we could combine the best of both worlds. So Vitis vinifera has the quality, seedlessness, cluster size, crisp texture, flavor, and color stability. Vitis vinifera has the disease resistance, and they also have these really interesting flavors that we could bring in. So if you could combine the two, they would be a super grape. But of course, like I said earlier, they have this difference in chromosome number, and they're just so long ago diverged that it's actually difficult to cross them. As people have understood that it would be valuable to cross muscadine grapes for the longest time. For over 100 years, people have been trying and working to develop these hybrids. Charles Deering and Louis Detchen were both breeders in North Carolina in the 1910s, and they made these really reciprocal crosses. They did a lot of very interesting work. It's kind of cool to read these papers from 100 years ago and see how interesting and how similar some of the things they're doing are to today. And they developed some initial F1 hybrids that had slightly better than zero fertility. So they're the F1 hybrids, like a mule, are largely sterile. So Deering developed this hybrid NCB 450 and Detchen developed this hybrid NC 615. And those had some level of low fertility. Those got in the hands after those programs ended of the three Bobs, Bob Farrell, Bob Zender, and Bob Dunstan. And they did a lot of work to further develop that material. Fair and received the material from NCB 450 and used that to make Fairer 30, which was subsequently used in the University of Florida breeding program to get Southern Home, which is a cultivar some may have heard of that has this unique leaf shape that was introgressed from Black Morocco, a Vitis vinifera grape. Bob Dunstan did a lot of work with back crossing to develop this DRX series of hybrids, and DRX 6040 has been particularly important as a bridge hybrid. At the same time, in California, on the Vitis side, Harold Olmo at UC Davis was working on understanding the cytogenetics of all of these initial hybrids and back crosses, and he developed some backcross hybrids in this E series he called that had fairly good fertility and wine quality. He also developed this bridge hybrid NC74 CO49-10 that has been used as a bridge to bring disease resistance from Vitis vinifera to bring disease resistance from muscadine to Vitis vinifera and seedlessness back into muscadines from Vitis. So at the same time, over hundreds of years, in like the 1980s and 1990s, Alain Bouquet in the INRA, the French program, had been working on developing Vitis type hybrids that had disease resistance from powdery mildew introgressed from muscadine. That's called this Run One locus. And those first three hybrids from his efforts over many, many years have finally been developed and released in France. They include Recibel, Reclia, and Rebellia. And his stuff has moved on to be used in the INRA program still, in programs in Crimea, in Hungary, and in the US in Cornell and other programs. At the same time, on the muscadine side, Jeff Bloodworth was a private breeder. He still is a private breeder. He works with Gardens Alive, and he had been kind of toiling on his own over many, many, many years to introgress seedlessness from Vitis vinifera into muscadine. He has a very interesting, innovative process using these bridge hybrids from Bob Dunstan and from Harold Olmo's program, crossing those to seedless vinifera, using the bridge hybrid as a female parent, and from there back crossing to develop these seedless muscadines that are, I think, really exciting products like Razzmatazz and Oh My and new ones in the pipeline. So I'll end with my presentation and turn over to Renee that, it's really cool to see over 100 years what people on the Vitis side and the Muscadinea side have been able to do in breeding these wide hybrids and working with muscadine grapes. We have Stenospermocarpic seedlessness introgressed from Vitis to Muscadinea. We have powdery mildew locus, powdery mildew resistance introgressed from muscadine to Vitis. But there are so many other interesting and complementary traits like seasonality, dry stem scar, color stability, flavors, all these cool things that we could be doing that haven't yet happened because of these difficulties of making these crosses. Also, the people who are breeding Vitis grapes have mostly worked in complete isolation from those breeding Muscadine grapes. So we haven't really done these like large scale collaborative projects in the past. We also haven't done anything using modern molecular and analytical tools to speed up what we're doing. So this has really created this opportunity that now is the right time to do a renewed collaborative effort and see what we can do applying teamwork and applying all of these new strategies and tools to speed up this process so we can get a lot more done in the next 100 years. So with that, I will turn it over to Renee.
All right, so thanks. Margaret did a really good job of just kind of covering and setting up the story of the next steps. And so, what I'm going to talk to you about is our project that has really come out of the relationships we've had and the history we have behind working with fruit breeding and in particular working with muscadine grapes. And so we actually, so I've been with the food science department for over 30 years. So I got to work. I knew uh, Jim Moore. I worked with Dr. Clark, and now I get to work with Dr. Worthington. And we have been for over a decade trying to get a group together to do research on muscadines with getting federal funding and getting a team together. But because it was such kind of a regional crop, like it's always been very hard, and we have spent a lot of time working to get a team together. And so before this project, we got a planning grant with Margaret and I leading that, so that we could help assemble the team. And so we really just been waiting for the right players to come along to have Dr. Worthington, who is so passionate about muscadine. I'm pretty sure that in her bottle as a baby, her mom stuck muscadine juice and just fed it to her as one of her drinks. And it's kind of part of her blood. So she's very passionate about it. And so after the planning grant, we submitted last April a full proposal to work as a team on the issue of muscadine hybrid breeding. And we waited for the call to see if we had gotten the project or not. And then in June, I got a call from the director, and we you have to realize this application is probably 100 pages long. Margaret and I worked on it and the team worked on it for a long time, months and months and months. And when he called me, I was waiting for him to say, are you funded? Are you not? Or if you are funded, what do you have to do to reduce the budget? But when he called, his but statement was, "You've been funded." And then he said, and then he didn't say but he said, "And I wanted you to know you were number one out of all of the proposals that we awarded." And they only awarded 17 proposals out of over 120 proposals. And so that just says something about the enthusiasm and the need and our team that we put together. One of the reviews of our team said basically that this was the who's of grape breeding and grape production in the United States. And so that's what I'm going to talk about right now. And Margaret can interject as needed, and if I get something wrong along the breeding program, because I'm a food scientist, so when she starts talking the breeding talk, I know a little bit, but enough to be dangerous. So the project is called Through the Grapevine, Developing Vitis by Muscadini Wide Hybrids for Enhanced Disease Resistance and Quality. And this is a 31-person project team from 12 institutions, and they're not all in the Southeast. You see the list on the screen, but we also have Cornell University, UC Davis, and USDA on here. And we're looking, and this award is from the USDA NIFA Specialty Crop Research Initiative, and we received $7.1 million for this project, and it's a 4-year project, and it just started in September a month and a half ago now. It seems already time is flying away fast. So Margaret spent a lot of time giving the background of the need for this. Grapes are a very high valued crop. The muscadine sector really isn't a big sector of that. It's, most of the industry is probably in the California and Washington and Oregon. I will add that in terms of economic value in the United States by state, the number 2 state now is actually Texas. So you have California as number 1, Texas as number 2, and New York is number 3. So we have a nice southern state coming in. They have over 500 wineries in their state, and so it's not always about you, California. I'm sorry if there's California people on here, but we do have some pretty cool things going on east of the Continental Divide. Um, and so we never really had formal collaborations in between Vitis and Muscadinea. So not only do we not have collaborations within Muscadinea that were large, the communities between the people doing research on Vitis have not really been working with the Muscadinea. So as part of our project, one of the things we did was we are partnering with another project called Vitis Gen, and their focus is really on Vitis type hybrids. So they're also doing hybrid and crossing, but they're really looking at those hybrids that are on the Vitis side. I don't know if you guys have ever had Dr. Andy Walker on talking to you guys, but he was a fruit breeder from UC Davis, and right before he retired, one of the things he was working on was hybrid crosses of vinifera with species that were Pierce's disease resistant. And that's something that, particularly if it's not Pierce's disease resistant, you can't grow it in Texas or in the South that are very far south. And so before he retired, he released 3 reds and 2 white cultivars, and he was really looking at a lot of some of our native species and combining some of those properties with that to give us some Pierce's disease resistant fruit. Um, but what we want to look at, and Dr. Worthington talked a lot about this enhanced disease resistant and fruit quality, and that's kind of one of our goals. And she showed a little bit of this and on her slide presentation, where you have the vinifera with these 38 chromosomes and these muscadinea with 40 chromosomes, and you've got all these attributes that we really like that are positive in vinifera that we would like to see over in muscadinea, and vice versa. Muscadinea, we've got cool flavors. We have really great disease resistance and a lot of the novel large fruit size and also cracking resistance. So we've got a lot of positive things that can go to vinifera and vice versa from vinifera into muscadinea that we want to start seeing. And when in her image, she had a super fruit in the middle of the circle, and that's kind of what we're thinking is, what are we doing in this project? Our goal is to develop a muscadine type hybrids that are going to be new fruit available in the market. Margaret didn't really talk about timelines, but fruit breeders are not in what we call a short game. They're in for the long game. And so they're not thinking, oh, I'm going to have something out next year. It just takes a long time. Some fruits and are shorter than others, but I'd say a grape is probably a 10 to 20 year in the breeding from first crosses to a release. So this is definitely a long game, but we already have things in the pipeline. The other thing Margaret didn't mention, because I always like to add it in, is that we are doing traditional fruit breeding using marker assisted technology. So we're looking for markers that have some phenotypes that are of interest for the breeding program, but we're not doing genetically modified work in terms of breeding. So I do want to make that clear that the work we're doing is traditional fruit breeding with marker assisted to speed up the pipeline of what's happening. So this is our project team. And so what is unique about our project team is it goes from beginning to end. When I say beginning to end, what I'm talking about is we have 6 main objectives, and those objectives go from the genetics with Chi Sun at Cornell University, breeding. Margaret Worthington is leading that effort. And then on to the plant pathology, where we have Lance Cladle-Davison with the USDA. He's also on the Vitis Gen 3 team, as is Chi, I believe. Then I'm working on the quality aspect. So Margaret and I co-lead the grant, but we also have leads within on the project. And then we're going on into marketing. with Melinda Newth at North Carolina State University. And why is that important? Because if we're trying to get out new crops or new products, or new flavors or muscadine flavor into vinifera, do consumers like that? Is it something like, I like to compare it to cilantro. You have your cilantro haters and your cilantro lovers. So are these flavors that are unique to muscadine? Is that something consumers want? So our marketing objective is going to look at that, and we're using a lot of examples on Fresh Market, but this does go into processing. It's just easier to explain, I think, to the public about it because they understand going to the store and buying cotton candy, grapes, and those things on the shelf because you can see it. With wine and processing and juice, you're doing a bunch of different things in order to get to that point. But there are attributes on the juice and wine production that are coming from vinifera that could go to muscadina that we could really use. And then the final objective is production with Dr. Mark Hoffman at North Carolina State University. So we're going to be working with growers. We're going to have plots. We have plots in several of our participating states. And we're going to be, we have replicated trials of the breeding program, but we also have some demonstration plots where we're going to be having field days and working with growers and looking at a lot of what growers think about new crops and this types of products that come out from that. So not gonna you don't really have to read this whole thing, but in our grant review, they're like, show us how all these things are working together. But we really do have kind of our three different teams are working together with each other, and then we cross over very much so. I think Margaret is on almost every team. She overcommitted, I think, but I think that the genetics and the breeding and the pathology are very aligned with each other, and they have to be kind of grouped together to address a lot of issues. And then marketing, quality, and production as well are kind of grouped together as a set of teams, but then also we have crossover. So that is a kind of our multi or transdisciplinary approach. But some of the work that we have already done, I'm not going to go over too much. There are these crosses and the hybridizations are being done and looking in particular at disease resistance. And we already have 2 releases out of seedless muscadina type hybrids. And that is Razzmatazz and Oh My, and I believe they've got a 3rd one that's coming out very soon. These were released in 2017 and 2019 by Gardens Alive. And the fruit breeder is a private fruit breeder, Jeff Bloodworth, who worked on these particular cultivars. So they're really, we're really starting to see some of these new fresh market muscadine cultivars come out, and they are having some of the attributes that we're thinking about on the breeding side. I was really lucky this year because I got to we went down to the fruit research station in Margaret's photo. She's got the whole fruit research station showing behind her here. And what's really neat about that is that it's about an hour and a half from where we are, but we were able to go down there. We were doing a photo shoot for the grant, and it just so happened to align with harvest of muscadine. And she talked about the freeze that we had in 21 and 22, and so we really haven't got to taste and have those fruit to taste them. And so when we were down there, we really got to taste the fruit and to see some of the crisp textures that she has and some of the seedlessness that's coming out. On the and then we've got objectives, ongoing activities also that are happening in Europe on the genetic side. I'm not going to tell. Margaret can hop on if she wants to make any comments about these, but and then on the pathology side, and I think what's really cool about the pathology side is, Lance Keitel Davis and Dr. Lance Keitel Davidson is really wonderful. I think he has a good grasp of what's happening in Vitis, and so linking all of his Vitis work that he's doing with the muscadina is going to really provide us with a good source of information. I probably can talk about quality a little more because it's my topic. So, but on the quality side, like I said, we're going to be looking at what do consumers think, what do about the fruit, what do they think about the juice, And what are those attributes that we can link to genetic markers to provide assistance for the breeders to help speed up the pipeline? So, and we are really focused on flavor. And we're doing a project very similar to this right now with blackberry, looking at interesting blackberry flavors that are coming out of programs, particularly in ours, but in other programs in the U.S. But we're going to do the same thing in muscadine. So we're going to look at these muscadine flavors and what the consumers think about them, and then help find those markers for the cool flavors that are out in these products. On the marketing side, we're going to be working with growers. We're going to be working with other aspects. And on the production side, we already have a lot of sites. And the thing about all these ongoing activities is that they were individual activities, but we weren't linked to anybody else. So now we're all linked together, and we'll be able to kind of move this slow train forward a little faster. And within each of the topics, we have sub-objectives. I'm not going to read every one of them, but genetics here, we're going to look at the cytogenic basis of these wide hybrids, success and marker transferability with the sub-objectives that are listed there. And then on objective 2, we're going to look at the hybrid populations and combining sensory and quality and disease resistance loci. On the pathology, we're looking at these loci and alleles associated with resistance to fungal and viral grapevine pathogens. And some of the things that we're looking at here are powdery and downy mildew resistance, black rot resistance, and those are some of the ones that we are definitely looking at identifying the markers for. On the quality, we're going to look at these key quality attributes affecting these preferences of these Vitis by Muscadine hybrids, and looking at all the marketing opportunities. One of the interesting marketing sub-objectives is we have someone that's looking at what happens when you kind of release a novel or new fruit into a market, and how is that impacted by the consumer reception of the product. I mean, think about when a consumer goes into the grocery store, typically you're going to see clusters of grapes in a bag or in a clamshell. Well, if muscadines don't come on a cluster and they're just sitting in there like, well, they kind of look like little plums, what is a consumer going to think about that? Is it something like where you see a new combined fruit that's a new name you've never seen before, or are they going to be willing to try it? And then, of course, I think we have a big extension component in our production element, where we'll be working with the growers, and we'll be delivering content all over the U.S., looking at the potential for the commercial production of these hybrids for the long-term range for our growers. And what do we expect to get out of this project? I always find this. Are we expecting 30 new cultivars in the first 4 years? Margaret will say no to that, I think. But would we like to see a couple? Yes. We already have, and that's only because we have some things in the pipeline already. But I think that this project, we have the ability to really accelerate the development of these hybrids, and with the focus on the disease resistance and the fruit quality. But we also need to develop strategies for our growers and our consumers on how to message and promote the cultivar throughout the supply chain. And that's a really interesting element. And we want to have more of the genetic computer vision phenotypic analysis that allows them to select elite seedlings early. So when they do a cross between 2 different plants, and they get 100 seedlings, and if we're looking for seedlessness, maybe that's 5 of those crosses, and we don't have to move forward. Whereas in the older days, we would have to wait until we had some fruit and see if it was seedless or not. But in this way, we can kind of skip some of those steps. And we really want to develop production guides for our growers. And overall in the U.S., this project is really about sustainability. So less inputs for our growers in terms of sprays and maintenance, where we can have more disease resistance and resiliency of our crops with changes that are happening with climate, and particularly in the United States, where we have hotter and more variable weather. We're seeing a lot more interested in the hybrid crops because they are more resilient than just the vinifera crops themselves. This is us when we took our photo. That's us throwing muscadines up in the air, and that's at the fruit research station. But that's the end of my presentation, and we would love to take some questions from everyone. And
Yeah.
yeah, let me stop share here. There we go.
Yeah, there you go. Thank you so much to both of you for the awesome presentation. It's probably one of the best we've had. And I have a few questions to kick this off, and go ahead and throw some questions in the Q&A, guys, if you have any questions. But um, one of my questions throughout all this, and I didn't see an updated range map, but are these Vitis and Muscadine hybrids, are they pushing the southern range further north? And if so, how much in terms of latitude, or how far north in the States can we
I'm gonna let Margaret answer that question, but I do want to add here that this year the USDA changed the cold hardiness zone
Oh
for
yeah.
Arkansas, and I know it shifted all over. So that's one thing to consider. And now I'll
Mhm.
let Margaret answer the question about muscadines.
Okay.
I will say, in my experience growing these kind of historical bridge hybrids from Bob Dunstan in the Olmo program, and the newer hybrids from Jeff Bloodworth's program, that we have a breeding agreement with them, so we're breeding with those materials. I have not seen any improvement at all in cold hardiness.
Oh.
I think one of the issues that I hope that we can address with some of these, using the molecular tools and stuff to guide what we're doing in our breeding. So far, anything that we're seeing, because it's been so focused on back crossing, either back crossing over generation and generation to muscadine, or vice versa, back crossing to Vitis, really the only characters that we've seen brought in from one to the other
Yeah.
have been these dominant characters.
Um,
So either it could be something that, like, this cold hardiness is very polygenic and it's not controlled by just one gene or whatever, that it's hard to back cross in, or it could be something recessive that it controls this like hardiness of the vascular tissue. There certainly is variation within muscadines, and I think we can make some improvements. One, we have not done any wide crosses that I am aware of muscadines to like Vitis labrusca or any of the really cold hardy American grape species. It's been to
yeah.
Vitis vinifera, to these table grapes mostly. And then within muscadines, I think we can improve cold hardiness by crossing with wild germplasm that comes from different parts of the natural range, crossing with more material from like West Virginia, Arkansas, Missouri, all those
Yeah.
kind of areas. So we
Right.
are kind of approaching it in both ways because it's a major objective for our program. And I guess also I'll say from North Carolina and in the Appalachian Mountains, because things that were found kind of high in the mountains might be more cold hardy.
And
But
Margaret, you've got a
no progress yet.
No progress, right? So are you? So are there? So you went. So in one of the slides that you went on botanical excursions or expeditions to find wild accessions or um, uh, species out there in terms in Texas and Arkansas, Tennessee. So is that happening in terms of like the Blue Ridge Mountains or some of these some of these more mountainous Appalachia areas that might have muscadines a little farther up in the elevation map?
We did that for a graduate student project. Just, I think no one had really done a very good study to look at how close, is the greatest diversity for muscadine grapes?
Yeah.
How closely related are these wild muscadines in Arkansas to these that you find in Louisiana, to those in Florida and North Carolina?
Right.
And so that was the primary goal of that experiment. We are crossing with some of the materials that we found from those
Okay.
explorations. Um, as far as collecting in the mountains of North Carolina. I would love to do that because it's a beautiful place to travel. But
Yeah.
I think I have not done a ton of that yet. There is a private breeder, Chuck Blevin, who has these like cold hardy Catalan Muscadine grapes. I have not tried them. I have not been offered the opportunity to trial them at our
Mhm.
station. I would be interested to trial them and
And
see how
he's also written a
they
book, I
Carlos.
believe, about this particular breeding endeavor, I think.
Oh, okay.
So you do have a question in the chat. Not in the chat, but
Yeah, but
Margaret, you have. Oh, you're not going to answer that.
I'm the host, so
Okay.
I can be selfish and ask you my questions
Yeah.
first. But so just one more thing. This kill event that you or selection opportunity event, as you so eloquently put, this bad thing that we would probably all be sad about. 85% of the of your your vines died.
Well, they were killed to the ground.
Or
That we had
hills, yeah.
suckers that came up.
Oh, so they killed to the ground, they came back up. So this
Mhm.
helps you in your selection process. Um, are you are you looking then like Cornell? Cornell's like way up in terms of latitude and stuff. Is there genetic material moving around and being grown at these partner or collaborative academic sites in different states, or is it just mostly the South?
Definitely, we have partners in Kentucky and in West Virginia in this partnership who will be growing some of the muscadine type hybrids. We have partners at Cornell, and I could send them muscadine grapes to die, but I'm not sure what the point would be. I think
Oh, I got
they
what you're
should
saying. Okay.
we'll wait until we have something that survives consistently in Arkansas and West Virginia and Kentucky, and then we'll take it up to New York.
Okay. Well, I do. Okay, so because I'm like, I want these grapes, and I'm a little farther north. So, okay, so let's get to the chat. So one of the first questions is roughly what percentage of Vitis and Muscadina F1 hybrids have sufficient fertility to proceed to the next generation, and does this change based on the Vitis or Muscadine species or cultivar that is used?
Well, that is a very good question. I find a lot of the F1 hybrids that I have looked at. I have not actually ever made an F1 cross myself. You know, I've kind of received some of these hybrids and they make a few grapes. So they have this low level fertility. A lot of them have a few grapes and then like you have to have the seed actually germinate from the grape. But I think a lot of them have some low level fertility. So first of all, that a lot of that work was done by these guys in like the 19 teens. You know, they did these crosses, Detjen made crosses, reciprocal crosses with muscadines, and a bunch of wild Vitis species. And there is a unilateral incompatibility. So you cannot make a cross muscat with muscadine as the female parent and Vitis as the male parent. And that
Okay.
was one of the actual barriers to bringing seedlessness from Vitis vinifera to muscadine is that caused the logistical hassle. It's kind of something about like the pollen doesn't germinate and grow down the tube effectively. I'm not sure exactly the genetic cause of it, but there is a unilateral incompatibility. But that's an excellent thing to do when you're thinking about making a wide hybrid cross, if that's something that someone wants to try, to throw in a bunch of different pollen parents and kind of see what sticks.
Yeah. So since our organization is a lot of fruit growers, but we're all like home orchardists and home backyard growers, and a lot of people do can't leave well enough alone. They've got to do their own crosses or create new things. It seems like a lot of work the foundation was really set by a lot of early citizen scientists in
Yeah,
this.
I actually think this really cool thing about this project, particularly for your members, that the three
Yeah.
Bobs did so much really cool work. And even Jeff Bloodworth just was an independent breeder. He didn't work for a company. His program was bought by Gardens Alive
Oh wow.
like several years ago, but he was kind of his own company for so long, you know?
Yeah. So would you almost encourage some of these other species that you named, like Lambrusca and so forth, just be like, hey, go ahead and cross and see what happens.
I think it's fun, you know,
Or try it, I guess, if you can
yeah,
happen.
I think, you know, for those who have the time and the passion, it's a exciting thing to work on making wide hybrids. It's kind of a fun
Cool.
challenge.
Cool. Did you, I try to figure out this question. Have you used embryo rescue in your breeding efforts?
Okay. We have done a little bit of embryo rescue, but when breeding grapes here in the past, if if you want to make a seedless cross, you have to do embryo rescue.
Mm,
For me, the economics work out where it's better for us to make seeded by seedless crosses and
seedless.
use a marker to find which ones are going to be seedless relative to doing a seedless cross and actually having to set up a whole embryo rescue and tissue culture lab. Another
Mm,
barrier to us in muscadines are difficult to grow in tissue culture relative to table grapes. They're also difficult to propagate. So it's very easy to propagate a Vitis vinifera from taking dormant cuttings in the winter, and muscadines, they propagate very poorly with dormant cuttings. It can be done, but
mm,
not on an economical scale. And I've heard
mm.
similar things in tissue culture that they are tricky species to work with. So we prefer to do seeded females by seedless perfect flower and use markers to sort out the progeny from there. That works out better in terms of economics for our program.
So you said that there's been an effort with international fruit genetics, the grapery. I think some of us are probably familiar with like Melissa's. That's a company that seems to introduce some interesting berries and fruits. So with the consumer saying that from one of your slides that there was an email address and nothing came back about the shape or size or color of the fruit. It was a lot of just feedback about the flavor. Do you feel that some of these grapes that might be released will eventually make them their way to like IFG or the grapery down the road so other people in the United States will get to try this outside of a regional area?
Oh, the muscadines?
The specific ones that you are potentially going to release next year or
Well,
as
probably not the ones that I am gonna release next year. I would love that. But I think from my experience with the US grape market, people have little interest in seeded material. I see a comment from here somebody who thinks that seeded material is not the seedless material is flavorless. I have not found that to be the case at all working in our program. I would encourage that person to try Jupiter. Order a Jupiter grape or a tickled pink grape. Google those because they are so flavorful and so good, and they're seedless material developed from our program here in the table grape side, and they are wonderful. So I think you can get grape flavor and seedlessness. I don't think that in a grocery store grape, there's any appetite for more seeded material out there in the US. I do think that there's an opportunity for muscadines to be a commodity on a supermarket shelf. I would love that. That is my
yeah.
dream for by the time I retire, for
Okay.
muscadines to be in between the berries and the grapes. They're such a terrific product. They're so flavorful, they're so interesting, and they're so good for growers in the Southeast US in terms of seasonality and opportunity. You've got your berries like blueberries and blackberries that come
Yeah.
on in June. When those growers really like need something to do with their labor, then you have a
Yeah.
fall crop that comes on. Muscadines ripen in September here and August
Okay.
further south. So I think there's tons of opportunities, but we need to have a coherent marketing strategy. We need to have seedlessness, flavor, dry stem scar, and texture all combined. And most importantly, you need a supply chain that can provide product 52 weeks a year. Otherwise, it will never be in the grocery store. And but already I think that they're a great opportunity for small scale growers, a farmers market, homeowners, that sort of thing.
Yeah,
So
and one thing I want to add here is because we didn't really talk about defining flavor, but when we're talking about flavor, we're not talking about sweetness and sourness. Like all fruit has that. We're talking about those unique, like the cotton candy like or yeah, the rose flavors,
yeah.
floral, foxy notes. We're talking about those other things. And I saw it in the chat. There's someone talking about, when things have strange names, they're not a big fan of it, but there are other products out there that where they're infusing flavors, like they're adding it after the fact. But we're talking about genetic flavor, not
Yeah.
not we're adding flavor to a grape after it's been harvested. But these are different unique flavors other than sweetness and sourness that would drive it. And to follow up with what Margaret said about 52 weeks out of the year to have a market. If you think about where the blackberry industry was 10 years ago,
Oh, yeah.
we were nowhere near every week you went to the market and saw a blackberry, but we're there now, right? So you have to
And
think
the growth
about
of the
it.
Mexican industry has really been a big driver of that.
Right.
The Mexican industry has been a big driver of the blackberries that you've developed.
And
Well, not necessarily our blackberries that we've developed, but blackberries in general. The thing is that you need to have a supply in the winter. If the US market cannot, if the US growers cannot supply in the winter, then it needs to come from somewhere.
Okay.
And the development of low chill blackberries coming from Mexico in the winter has been a real benefit to growers in the US,
Okay.
particularly growers
consumers.
in our part of the country,
Hmm.
because they're their rainy season comes on like June through November, when is the time that blackberries are produced here. So it ends up
Yeah.
being a good complement. But it's hard to get a spot. If they'll stick more apples in.
Okay.
If you can't supply
So
52
you're
weeks a year.
competing for a
Yeah,
lot of valuable space.
for sure.
Um, but you said in a slide that there might be some muscadines that are with thinner skin that might be hitting the market soon.
I think already there are muscadines that have so much better texture than some of these old school. I mean, I love the wild muscadines for their flavor, but the
Mhm.
texture is, you
Yeah,
have to spit that skin out.
yeah,
And
that's a
skin
thick
is
skin.
where like all the nutrients and the flavor are. I mean, we want to eat the skin. So things like Supreme. Supreme is a pistillate variety, so it needs a pollinizer, but I think it's a terrific grape that's already out there on the market. Big size. Ruby Crisp is a release out of the University of Georgia program. Patrick Connor, the breeder at University of Georgia, is wonderful. He's a great collaborator and a wonderful breeder, and he has Ruby Crisp that is, I think, difficult to grow commercially in terms of yield and some other quality attributes, but it's got great texture and good flavor.
Oh, cool. So we might get because I think usually up north we find like the muscadines and so forth available in specialty stores like Whole Foods or Trader Joe's sometimes. Um, and you have to,
Margaret, where that's the one thing you didn't mention. Like if for our releases from the U of A, where would they be able to find them next year?
Oh,
yeah,
we'll have them for sale at I think Ison's for sure. Bottoms is propagating them. And Greenleaf Nursery in Oklahoma. It's kind of a we've been talking to tissue culture partners and to Double A, but Double A I think has had trouble with muscadines in the past because they
okay.
are sensitive. It's hard to grow up in New York. So
And the other question we get a lot is like they'll want to just call or email Margaret for a plant or two, but it really doesn't work that way.
Yeah,
So we do have we have growers that want to work with us, but there's a process that we have to go through, and
right.
it's usually people we're approaching to work with, not
Okay.
vice versa. But if you're just looking for a single plant or a home plant, the nurseries that we are going to be working with is where you would be purchasing them,
and they
and they
cater a lot
probably
to the
won't
small
be,
growers and homeowners.
yeah, and they probably won't be out at Lowe's and Home Depot's out for a few years. Like it will not be that, not right away.
Not that common. And it also will be very it'll also be very zone specific is what I've learned from this talk.
Yeah.
So it'll fit for our southern growers a little bit more. Okay, and then I think that's it. Oh wait, is there anything in the chat? Is there any nutrient variance in the fruit that you've developed versus like the bunch grapes or anything? Is there enhanced
Well,
nutrition?
in the history of grapes, muscadine grapes have a little bit, I won't say nutrient, I'd say nutraceutical or healthful compound history. But in general, dark fruited varieties have more than light fruited varieties, just like any other fruit. But I don't know about like when we start breeding, it will be something probably we're going to be maybe in the future looking at, but it's not part of this grant that we're working on now.
Yeah,
Okay.
and I'll say, muscadines, like Renee said, they have been known to be a healthful fruit that has lots of like anthocyanins and
Resveratrol,
other,
phenolic compounds.
yeah, other
Mhm.
phenolic compounds and flavonoids that might be important for health. There are nutraceutical products that are made with muscadine seed extract and also the kind of skin that is left over from the processing of wine. So you could look up muscadine nutraceutical compounds online.
Yeah. We're going to be working, one of the part of our quality objective, we're going to be working with Mighty Muscadine out of North Carolina to kind of distribute a juice out to people in the U.S. at different events that we're going to be doing,
Oh,
and with
cool.
a QR code so you can give us your input about what you like or dislike about a juice. So maybe we could partner with you guys to work with your members.
Yeah, you can. Yeah, I'm always wanting for free stuff to eat or drink, so that's count me in.
All right.
Um, with this marketing that you're doing, and you're also doing the wine part of this, Renee. Are you fine? Are you is are just humor me here because I haven't grown up in the South or anything. Are there are there wines that are based upon just muscadine grapes?
Absolutely. One of the
Oh,
number one sellers in Arkansas, I'll name a name, is a Blue
okay.
Parachute out of Post Winery. It's 100% Carlos, and it's they're the number one seller probably. I know for them it's their number one seller. It has a big blue parachute on the label, and it's a lighter alcohol, so it's like 8% alcohol, 7 to 8%, and
Okay.
it's slightly sparkling,
Oh,
frizzante.
cool.
But a lot of our wineries, and I'm not just in Arkansas, have 100% muscadine juice or muscadine wine. And it'll usually, sometimes it'll have a simple name like red muscadine wine or white muscadine wine. But yes, definitely we do have that available in all the kind of southern states: Florida, North Carolina, Georgia, Arkansas.
Oh, okay. So I'm not in touch with
Yeah,
the
you can actually, you can go to the websites of some of the bigger Post Winery, Whitaker Winery, at least on the Arkansas side. Canandaigua, that's not right. There's a winery, Lakeview Winery in Florida is like has the highest volume of muscadine wine production in Florida.
Oh my gosh. Okay, cool.
But you can buy those online and have them shipped to yourself. If you,
Oh
for
wow.
those,
Okay.
I challenge everyone, the people that didn't respond, that's responded not yet, to purchase a bottle of muscadine juice or wine to try so that you can next time answer yes.
I think we've got one of our members on here who loves wine, Eric Bina, who also lives in Florida sometimes. So I'm not sure, Eric, have you had muscadine wine? You could drop that in the chat as we come to an end here. And I think that's it. This is super exciting to follow this work and where you end up in terms of the combination of the hybridization of these two grape species. And we're lucky to have people like you doing this work. And thank you for taking the time to inform our membership. Someone said in the chat that some of these early breeders were probably, or mid-century, were probably NAFEX members back in the past. And so it's kind of cool to see these backyard citizen scientists doing these things and working to help you guys out in your academic settings. So keep reading it out there, fellow members of NAFEX. Uh, yeah, keep combining things.
And feel free to contact us if you have any questions. And
Oh, you're already going to be so everyone, Margaret also works with the berries, the blackberries in Arkansas. So we're most likely going to have her on for a talk again because these are your Primark, your Natchez, your Ponca, all these other ones that you've heard of, you've tried, the Primocanes, the Floricanes. So a lot of us want to get smart on that. So yes, we will be hitting you up again for to get smart from you. So with that, I don't see any more questions for our speakers. So I want to thank you both for taking the time to speak to us. You are both now new NAFEX members for the next year. Yeah, so that gets you into all of our talks that we've recorded, as well as like a lot of the history of the Pomonas, our quarterly publications that go back for to mid-century. So that might help you out if you're looking for some research notes or something like that. And I'm just going to read this closing statement, and then I'm going to let everyone go. But on behalf of NAFEX, I would like to thank our speakers that made this a great session for our last session during this week. I'd like to also thank our sponsors, One Green World, Copenhagen Farm Nursery, and Southern Cultured Orchard. I'd also like to remind everyone that this talk or this session is being recorded and will be available on nafex.org, our webpage, in a few days. And it will also be available 1 year from now on our NAFEX TV YouTube channel. There'll also be other downloadable content. So don't miss the grower meetup tonight, which is our show and tell. Come with a drink or come with something to show us, or both. And that concludes our session for the evening. So I'd like to thank you both once again for really just informing us so much about all these efforts that we would not have known about, honestly, if it wasn't for the two of you. So keep it up, and we'll be in touch.
Thanks for having us.
Thanks, everyone. Have a great night. See you soon.